Welcome Junior Techies to this video where we are going to be looking at a typical investigation question, and this one particularly is about is CO2 produced by living organisms during cellular respiration. So it's one of those investigative questions that you see there on the front of this. So do stick with me while we work through this investigation together. My name is Mr. P. I'm dealing with the theory and some of the practice questions of this section on cellular respiration. So let's dive right in and see if we can better our understanding within this area. Right, so let's look quickly. This investigation is one that deals with carbon dioxide as a product of cellular respiration. Now, we have already started studying this section, so we should already know a little bit about what's going to be happening here. So this should not all be foreign to us when we do our analysis here quickly. So this investigation is looking at living organisms that produce carbon dioxide. But how do we know that it is carbon dioxide that is being produced as a byproduct of aerobic respiration? Now, just some context, because that's what this first paragraph here is giving us. We can test for the presence of carbon dioxide by using clear lime water, which does turn milky when carbon dioxide is bubbled through it. Now, that's one of those tests that you would have learnt about in your life sciences career, and this is a fantastic application of that little bit of science. You see there that there's made mention of soda lime, which is used to remove carbon dioxide from the atmosphere. So in other words, that soda lime absorbs the carbon dioxide as a gas from the atmosphere. And then we've also got sodium hydroxide in a solution, which actually removes carbon dioxide in liquid as well. So there's a bit of context for us. So what the soda lime and what the sodium hydroxide can be used for. And you'll see this when I show you the experimental setup in a little bit. And what we do now is that we use that little bit of science there to make sure that there is no carbon dioxide entering the experiment before cellular respiration takes place. This would mean that we only test for carbon dioxide that can be produced through cellular respiration. Okay, and that would be by the organism that we select for this specific investigation. So our hypothesis here is that carbon dioxide is produced by living organisms during respiration or cellular respiration, and our aim is to prove it, that it is released by living organisms. So let's quickly have a look by highlighting a few of the important points here. Milky color of lime water is a positive test for carbon dioxide. You'll see there is soda lime and sodium hydroxide, which I've highlighted already. So those are important points to remember when you are understanding why these specific substances are used in this investigation. Okay, and there you can see it removes and it removes. So that's important for us there. Some of the materials or the apparatus, small organism, so this is that respiring organism. Alcohol to make sure that our apparatus is clean, because the last thing you want is you want a microorganism to add any type of respiration to your experiment. So we make sure that we clean all of the surfaces in the apparatus with alcohol to kill off all of the microorganisms that may be present on those surfaces so that they don't interfere with the investigation at all. We have our large jar in which we're going to place our organism. 3 test tubes. These are for our solutions, and you'll see which solutions I'm referring to now. Rubber stoppers to make sure that each of those are sealed off quite nicely. We've got some glass tubes for the transport of the gases. An electric pump, this is for the movement of the gas through the entire system. My soda lime and sodium hydroxide, we now know, are to remove the carbon dioxide from the experiment. And then my lime water for our CO2 test. Great. Let's have a look quickly at our method. We're going to use a small organism, and in this case it's a snail. We're going to sterilize our equipment so it is not contaminated by microorganisms. We place the snail into a large jar, just enough space for it to respire. And we're going to set up the apparatus as shown in the figure, which I will jump across to in a minute. We're going to give it some time. So we've got some test tubes, and we're going to allow the experiment to take place. So let's have a look at our image quickly. Okay, so here we can see we've got incoming air into my system. I have soda lime at the onset, and what the soda lime does, yes, it removes the CO2 from the atmospheric, or should I say from the air that enters the system. Okay, I have my sodium hydroxide solution through which I am going to bubble the air as well. So there's a double control there for the removal of CO2, which means the gases that enter that first boiling tube or that first test tube and that is going to exit through the glass tube Y is not going to have any carbon dioxide in it. And how we test for that is we use the lime water to make doubly sure. That's the first test for CO2. In test tube B, I'm bubbling all of the gas entering my system through lime water first to make sure that there's no CO2 present. Do we expect the lime water in test tube B to turn milky? No, we don't, because we've removed the CO2 through the use of soda lime as well as the sodium hydroxide solution. That means that the air, the gas entering the bell jar or the jar C is not going to have any CO2 present in it at all. We have a snail in jar C that is going to respire, and what we expect here is the use of oxygen and the production of CO2 within jar C. That means the air leaving jar C and entering the test tube D, we expect that to have trace amounts or amounts of CO2 in it. So in the test tube D, the lime water should turn milky if my snail was respiring and producing carbon dioxide in this experiment. All right, now I'm going to ask you a question very quickly. Looking at this experiment over here, do you think that this is a valid, reliable experiment in the testing for whether or not CO2 is produced during respiration or aerobic respiration? What you could maybe do is on a piece of paper, jot down a few points that you maybe don't agree with in this investigation, and then try and redesign the experiment to make sure that the validity of the experiment is improved upon. If we look at the results here, test tube B, the lime water remains clear because carbon dioxide is removed by both the soda lime and sodium hydroxide at the onset of this experiment. Test tube D, lime water will turn milky because carbon dioxide is produced by the snail through aerobic respiration. And in conclusion, because of the results in test tube D, carbon dioxide is produced during cellular respiration of living organisms, and in this case that would be the snail. Right, Junior Tikkies, thanks for joining me in the discussion of this investigation. I hope it's given you some insights and some valuable tools in experimental design. One last question I'm going to leave you with is, how would you improve the reliability of this conclusion that carbon dioxide is produced during respiration by living organisms? How would we improve the reliability of an investigation like this? Well, I'm hoping you're going to be thinking of repeating the investigation again, and then potentially, if we look at the living organism that we've used here, is maybe rotate through a few different living organisms for bell jar C. So we are increasing our sample size, or we are going to repeat the investigation. All right, Junior Tikkies, thanks again. I will look forward to seeing you in some of my future videos. Please make sure you get a hold of the worksheet as well as the memo and work through those questions to make sure you understand exactly what is needed and exactly how to do a question analysis for your final exam to get the best possible grades. the end of the day. Right, have a good afternoon, Junior Turkeys.